Okay, hello everybody, and welcome back to part three of chapter 27. And uh, this will be the last part of the chapter 27 explanation or lecture discussion, you can call it. So let's jump right in. You might remember that on at the end of part two, we finished up with gram-positive bacteria. And so we finished up with all of the bacteria during that session. And so today we're going to cover the archaea. And remember that the archaea are a domain of living things, so it's bigger even than a kingdom of living things. So this domain archaea, they do share certain traits with bacteria, but they share other traits though with eukaryotes as well. And then also they have a few unique characteristics that only they have. Remember, here's the phylogeny, the proposed phylogeny for the relationship between the three domains, eukarya, archaea, and bacteria. And so here are the different characteristics of the three domains. Whether or not they have a nuclear envelope, whether or not they have membrane-enclosed organelles, or peptidoglycan in cell wall, etc. Whether or not they have a sucrachromosome. And you can see that basically now on here, the membrane lipids are very different. Archaea are very different from the two other domains in terms of membrane lipids. The other domains have unbranched hydrocarbons as membrane lipids, but the archaea, though, in their membranes, they have some branched hydrocarbons. And also, in terms of being able to grow and survive at temperatures over 100 degrees Celsius. And remember, water boils at 100 degrees Celsius. So those that are able to grow at temperatures over 100 degrees Celsius, there is no bacteria that's able to grow over 100 degrees Celsius. And of course, there's no eukarya that are able to grow at that temperature. But there are some thermophiles, they call them, ones that really like to be in high heat environments. So some of the archaeans can do that, very few. And remember, domain eukarya is all of the eukaryotes. So all of the protists, all of the fungi, all of the plants, and all of the animal. Okay, so protist kingdom, fungus kingdom, plant kingdom, and animal kingdom are all eukaryotes. So on with the archaea then. So some of these archaea, as we just mentioned, live in extreme environments. They call them extremophiles. Phile usually means loving. So extreme halophiles is high salt content. Extreme thermophiles means they like to live in an area that's very hot, because thermo means heat, like a thermometer measures the amount of heat in a certain environment. And so this orange color and kind of green color produced in this very hot spring in Yellowstone, by the way, very hot spring. That's produced by all the archaeans, extreme thermophiles that are able to grow in there. And then there are also things called methanogens. So methano refers to methane, and gen, remember, means to produce or to generate. So they produce methane. They're in swamps and marshes, and sloughs as well, and produce methane as a waste product, and lots of methane. And these methanogens are strict anaerobes, so they must be in an environment without oxygen, because oxygen is actually poison to them. The O2, the oxygen, is actually poison to them. Let's see. So Prokaryotes then play crucial roles in the biosphere. So now we're going to look at prokaryotes in general, bacteria and archaea. They play crucial, important roles in the biosphere. The biosphere is basically all life on Earth, the sum total of all life on Earth, because bio refers to life and sphere refers to the whole Earth. And so they're important. They're so important that if they were to disappear, other life probably would not survive on Earth. So they play a major role in recycling of chemical elements between living and non-living parts of the environment. And so the chemoheterotrophs. Remember, chemo is chemical, hetero is other, and troph is feeding. The chemoheterotrophs then are decomposers, and they break down dead organisms or parts of organisms, and also you break down waste products as well, so they can be recycled. The nutrients can be recycled through the environment and the ecosystem. They can convert some molecules to forms that can be taken up, forms that can be more easily taken up and used by other organisms. And for example, in part 2, we saw that some of these prokaryotes can convert nitrogen, take nitrogen out of the air and put it into a form that can be used by plants. Under some conditions, prokaryotes can increase the availability of nutrients required for plant growth, like I just mentioned here with nitrogen, for example, or nutrients that are in the soil, iron and magnesium and calcium, and a lot of nutrients that are in the soil that need to be used by plants, accessed and used by plants. So in soil that had no bacteria, the plants didn't really grow as well as plants with different strains of bacteria in them. Now, but of course the big question is, well, okay, is this difference between here and here, or between here and here, is that difference significant though? So then prokaryotes can also immobilize or decrease the availability of nutrients by using them themselves rather than letting other species use them. Now let's talk about symbiosis, and we mentioned this a few times, and you probably remember this from high school biology. Symbiosis, then, a relationship between two different species. They live in close contact, and if one is a lot larger than the other, then the larger one is called a host, and we mentioned that term before in this chapter. The larger one is called a host, and the smaller one is called a symbiont. And if the symbiont lives inside the host, you call it endosymbiont, but we'll see that in just the next slide. And they often form symbiotic relationships with larger organisms because they're very small. And there's 3 different types of symbiosis then. There's mutualism, commensalism, and parasitism. So in mutualism, the relationship is mutual, basically. Both of the organisms benefit, both species benefit from the relationship. In commensalism, one benefits while the other is not harmed or affected in any way. In parasitism, though, parasites, in other words, so that's, of course, a parasite then causes harm to the other species and its host. Then, so parasite will harm the host but not kill the host. Actually, technically, in some instances, the parasite does actually kill the host, at least indirectly. And then parasites that cause disease are called pathogens. Because remember, pathos is disease, like pathological. Pathos is disease, and gen, of course, is to generate or to produce. So, like this fish that's parasitized by a number of prokaryotes. The existence of some ecosystems. So some ecosystems could not exist without different prokaryotes. In fact, the more they study, maybe no ecosystem could exist without some sort of prokaryotes. the ecological communities in the hydrothermal vents that are in deep areas of the ocean, they depend on chemoautotrophic bacteria for energy. Some prokaryotes are human pathogens, like we've seen, but others have positive interactions with humans. Because human intestines, believe it or not, don't want to scare anybody, but your intestine is called home to between 500 and 1,000 species of bacteria. But many of these are mutualists that you actually benefit from, like E. coli is a mutualist. And they break down food that our intestine cannot actually digest. So a better way to, I think that's a better way to phrase it. So they cause about half of all human diseases, and some bacterial diseases are transmitted by other living species, like Lyme disease, the rickettsia that cause Lyme disease. It's basically carried by ticks and transmitted by ticks. Carried and transmitted by ticks. And so in some of these ticks, like the deer ticks, can be very, very small. Almost just a small speck, and it's a spirochete that causes that. And in 2/3 of the cases, it leaves a red mark, but in about 1/3 of the cases, it doesn't leave a red mark. So just because there's no red mark doesn't mean you don't necessarily have Lyme disease from that tick bite. So these pathogenic disease-causing prokaryotes then cause disease by releasing exotoxins outside the body, or endotoxins that are produced inside the things. So exotoxins that are secreted to the outside of the prokaryote cell, and they can cause disease then, even if the prokaryotes themselves are no longer there. The endotoxins are released only when the bacteria die. Yeah, so the endotoxins are inside the bacterial cell, and they're released when the bacterial cell dies and the wall breaks down. Now, we've talked about this before, but horizontal gene transfer then can spread genes, but it can spread genes associated with virulence, that is, ability to cause disease. So pathogenic strains of E. coli, most E. coli that are in human colons are not pathogenic, but there are pathogenic strains. And those strains contain genes that were acquired through transduction, that getting them from another species of prokaryote. In experiments that have given experience with prokaryotes, then have given important advances in technology. E. coli is used in developing gene cloning technology, and the prokaryotic CRISPR-Cas9 system, remember, helps us alter genes in many different organisms. And bacteria can be used to make natural plastics. Better for the environment. And so here's an experiment where control cells and the experimental cells. And bacteria can be genetically engineered to produce ethanol from agricultural and municipal waste biomass, like switchgrass and corn. Here's some cells that do that. They can be used in bioremediation, using living things to remediate or restore parts of the environment by removing pollution. Some of them can eat oil that's been spilled into the ocean. For example, that's what you see here, then, an oil spill. And So remember, there's a wide variety of different prokaryotes. And then some of the thought questions that you can look at in the book. And I think that's pretty much it then for this chapter 27. So as always, though, let me know if you have any questions about this or any of the other materials or any of the parts of the course. So study through these PowerPoints, and if you like, listen to these lectures and discussions, verbal discussions that I'm putting up in Big Blue Button. Anyway, if anything comes up, just give me an email or a phone call, or you can even stop by the office as well. So until next time we'll be covering chapter 28, by the way, and that'll be on the Protus. So keep looking for that, because I'll be putting that up within probably 24 hours from now, January 29th. Thanks. Bye.